First, the eye. Charles Darwin himself said, "To this day, the eye makes me shudder." And even serious scientists have sometimes queried whether there's been enough time for the evolution of the eye.
What I'll do for you today is I'm going to talk about how, one, the eye shows extraordinary evidence of design. Two, I'm going to critique for you that evolutionary narrative.
Well, suppose we start with an ancestor who didn't really have an eye at all, but just a single, simple sheet of light-sensitive cells. So this animal, with hardly any eye at all, will at least be able to tell the difference between light and dark. The next stage in evolution would be to have a shallow cup. This animal would be able to tell the direction that light is coming from, because there a shadow would appear. A shadow would appear there. And if you can tell the direction a light is coming from, then you can tell the direction that a predator is coming from. That shadow effect that we've just been witnessing would work progressively and gradually as the cup gets bigger. Let's make it bigger still now, Bryson. And this cup is even more effective. And if we go on to the next stage where we make the cup gradually bigger again, so big that it becomes just a little hole in the end. Right now, this animal has a very good idea of exactly where the light is, and by the same token, exactly where, for example, a predator is. And I think with this eye, we might even get a little image. See if we can get an image of Bryson's hand. That smudge there is Bryson's hand, and you can just about see a very dim image of his fingers. So an animal with an eye like this would be able to see perhaps just a little bit what kind of predator it was. Go to the logical conclusion, which would be a pinhole. Remember, this is all gradual change in evolution. Right, let's see if we can see your hand again, Bryson. Now I can see a rather precise picture of Bryson's hand. It's not a very bright one, but I can see every finger clearly delimited. So I could see, if I were this animal, I could see my predator in some detail. Now, a pinhole camera is not a very good way of seeing. It does produce a sharp image, but because it's so narrow, you hardly get any light in. The answer to this problem is that ingenious device, the lens. How might the lens have evolved? Well, let's imagine that it started with just a single transparent sheet of some transparent material. And all that this was doing, it's not a lens yet, all that it's doing is protecting the eye. In Nautilus, seawater flows right inside the eye. This animal now has some protection. And the eye is really just the same as though there wasn't any transparent material there. Now, we're going to use an optician's set of lenses here. It would be nice to be able to have just one bit of transparent material which we would then squeeze and make thicker. But we can't do that, so we're going to replicate that effect by a whole series of little lenses. So this is the next stage in evolution. This animal here, let's get an image of that. Okay, that's a rather better and above all brighter image of the hand. Let's have the next lens in. Right, now, if an animal that had an eye like that would have a really very, very clear view of its world, it could tell exactly what its predator was.
And you ask them, well, how do you believe the eye came about? They would tell you this very nice story of how you have a photoreceptor that appeared, and then what happened eventually was that photoreceptor ended up inside of a kind of a cup, and that cup sort of had shadows, and eventually it turned into a pinhole, so it became maybe more of a well-focused shadow. Eventually you had some gelatinous material that may have acted like a lens, and eventually a real lens formed, and then eventually you get the modern eye, the camera-type eye that you see in vertebrates. Have, again, more engineers working with biologists, and what they found is that life looks like it's a top-down design system. Now, what do I mean by top-down design? Well, it's kind of like if you design a car, what you do is you look at the top level of the organizational hierarchy. You look at the major systems. So if you're going to design a car, you know, a car has to have an engine, it's got to have a passenger compartment, It's going to have a transmission system. That's the highest level in the organizational hierarchy. And then what you do is you design every major system in such a way to work with every other system with extreme efficiency. And every part of each subsystem is designed specifically with the overarching goal in mind. That's top-down design. That's what you see in life. You anticipate what's in the human visual system from the perspective of an engineer. And probably the best example, the best archetype would be that of a digital camera. Because if you look at a digital camera, how does it work? Well, a digital camera has to have a lens that will focus light onto some array of detectors, whatever they happen to be. And what happens is your lens typically will be in front of an aperture, which is a hole that can get bigger and larger and smaller. Now, why is that? Because if you're obviously in dark environments, you want a larger hole because it lets more light in. And that's helpful for a lighter image. But the problem is if you've got a bigger hole, that's going to result in an image that's a bit more blurry. That's why your vision is a bit more blurry at night, because your pupil is larger and you get more diffraction taking place. Well, the aperture gets smaller in bright light because, one, you want less light to come in so you don't flood your device and kind of wash everything out, but also it creates a sharper image. Well, what do you also see? You also find that the image has to be focused. And that could be because you might change the direction of the lens, you might change lenses. So whatever way you do it, you've got to have a way to alter the focus of your lens system to create a sharp image exactly where your photodetectors are. That's something you need to do. Well, also what's going to happen, you're going to have your photodetector array in some ways pre-process the image. It's got to remove noise, it's got to kind of sharpen edges and things. And then you go through and you will send it to some, if it's, let's say, a machine vision system, it then has to go through a complex process to reconstruct the image and identify it. That's sort of the pattern that we're going to use. That's the template we're going to use as we study the visual system. Before we embark on our journey of studying the eye, we want to again think in advance, how would we design the system as engineers? Well, first of all, you recognize that your system is going to have specific goals. The goals will be you have to detect the image, you have to map the image in the brain, you have to identify the object. So this is the goals of what our visual system will have to do. Well, that means there's certain objectives. You focus the light, you detect photons, you send signals to an image processor, and then you reconstruct the image. So we can expect to find each of these in our visual system. But also, as good engineers, is we recognize that if you're going to design a visual system, you've got to deal with constraints and various concerns. You have to deal with resolution, and that's going to be limited by the light wavelength and the eye size. So these are going to be very important in our resolution. Clarity. Whatever the light goes through, and they have to be cells, has to be transparent. So this is something that we're going to have to look at and how that happens. Coherence. What happens is when light goes through a spherical lens, you produce a very sharp image from the light that goes through the middle of the lens, but as you go out, as light goes through the outer edges of the lens, you get what's called spherical aberration, where it's going to be less focused. It's going to be fuzzier. It's not going to focus in the same way. So we're going to expect that our living system is going to somehow deal with spherical aberration. Another problem is chromatic aberration. What happens is when different wavelengths of light goes through a lens, it will focus at different points. So like red light is going to focus behind blue light. So that's going to create a fuzzier image with different colors. So we're going to expect that our visual system is going to deal with that. Now, as far as the detectors, there's going to be the issue of the photon frequency. So you have to have special photoreceptor molecules or detectors that detect the right frequency. So we're going to expect some very complex chemistry. Resolution and color. You're going to have a detector density and variety. So in order to get high resolution, you're going to expect to find an array of photoreceptors that are going to be small and tightly packed. That's our expectation. And what you're going to expect is you're going to have different photoreceptors that detect different colors in a tight matrix. So in any given part of your matrix of photoreceptors, of your grid, you're going to expect to see like red, blue, and green photoreceptors mixed in with each other so that you can detect different colors. Here's our expectation. Time resolution. you're going to have to reset your photoreceptor very, very quickly. Because what happens is whatever physical change takes place when this photon is detected, it has to reset so it can keep seeing things. Like, you ever have an experience where you look in a bright light and then you can't see for a while? So the eye has to somehow protect against that. You're going to have to deal with sensitivity range. In other words, the light difference that a terrestrial animal encounters is enormous. So if you're in, let's say, very dim light near dusk, you might have to detect single photons. But if you're in the bright sunny day, it could be 1 trillion times more bright. So your photodetector is going to have to somehow adapt so that you can deal with this huge range of intensities. Because if it just turned on and turned off, you're going to either be washed out or you're not going to see anything. So that's going to be a challenging problem to deal with. And then finally, you have to repair damage, because your photoreceptors after you're born are all you get. You don't produce more photoreceptors, so you're going to have to repair these guys. So these are issues we're going to be very concerned about as we start to embark in our study. So let's just jump right in. What do we see? Well, exactly what we expect. The cornea is the outer layer of the eye, and it's connected to the sclera. And what the cornea does is it protects the eye, and it partially focuses the light. You then have a lens which focuses the light on a very sharp image at what's called the fovea, which is in the retina. So here's your retina, and the retina is your array of photodetectors. And what happens is the center is the fovea in the macula, and that's where you have the highest density of photoreceptors, where you have like your center vision. So that's where you can see things very sharply. But the peripheral vision you can see, but it's not going to be as high resolution in there. Also, what's really fascinating is here again, what you see is 3 separate types of photoreceptors. You've got red, blue, and green, which all detect different wavelengths of light. Now, this is what's super cool. You're going to love this. What happens is if you look at, let's say, the green photoreceptor, there's a range of frequencies it can detect. It's not like it detects one frequency. It's like a bell curve. So what happens is a challenge that the system has is if you have, let's say, detect photons at the very peak of your curve, then you don't need a lot of photons and you'll detect it. But the problem is, if you detect, let's say, higher frequency or lower frequency, then what's going to happen is you'll need more photons to give you the same signal. So one photoreceptor can't tell if you're getting a few photons at the peak or lots of photons off the peak. So what happens is your eye can compare 2 separate photoreceptors, look at the input from both, do a complex calculation to determine both the intensity and the frequency of the light you're absorbing. And what's absolutely amazing is each of these photoreceptors, or sets of photoreceptors, go to what are called the ganglion cells. And those are the cells that will have nerves that go to your brain. And what happens is you are processing multiple photoreceptors at once, so you get a very complex image information that goes through each of those cells. It's extremely genius. Now, what you have in your eye is about 100 million what are called rod cells, and those are more in the periphery. And what it does black and white vision. It just sort of detects one frequency. It's not concerned about color. As you go closer and closer to the fovea, you have higher and higher concentrations of the color cells, because in the color cells is what you need to get the more detailed information for color vision. That's really, really key. So this is sort of the structure of your eye. Now, that's sort of the big picture, but let's kind of focus in a little bit more. What you have is you have a pupil, and the pupil is surrounded by the iris. And the iris is the colored portion of your eye, while the sclera is the whites of your eye. That's what you see. And what happens is you've got specialized muscles, and what these specialized muscles will do is it will cause your pupil to get smaller if you have bright light and larger if you have less light. But what's absolutely amazing, is those muscles are controlled by completely different parts of your nervous system. So one part of your nervous system will control the pupil getting bigger, Another part of your nervous system controls the pupil getting smaller. So if you think about the engineering behind that is astonishing, because what you have to have is you've got to have your photoreceptors, they detect the light, and somehow they have to detect the intensity, and they have to send a signal to the brain, and then the brain has to go through two completely separate parts of the nervous system to tell the pupil to get larger or smaller in order to have the right amount of light. Again, if you just had one set of muscles to get only larger or only smaller, your eye isn't going to work very well. Now, what's also interesting is if we go into more detail, what you have here, as we can see in this image, is you have the lens, and what the lens has is what's called the ciliary body. And what the ciliary body does is it has specialized muscles, and what these muscles do is they contract these ligaments, and they cause the lens to flatten or to be more bulgy, depending upon where an object is. So if your object is at a distance, the ligaments are pulled, they flatten the lens, so you get a very sharp image on your retina for things at a large distance. If you're looking at something closer, the ligaments will, the tension will release so that you can focus on something that's near. So here again, you have a feedback loop where your brain somehow can detect if an image is in focus, if it's out of focus, sends a signal to these muscles to pull it just the right amount to always produce a focused image. It's really quite amazing. Now, I talked about this problem of spherical aberration. What's amazing about the lens is the lens actually has a different index of refraction in different parts of the lens. So as you go from the center out, the index changes, and that change of the index of refraction, according to a specific mathematical relationship, is what eliminates spherical aberration, which is really pretty astonishing. Also, I mentioned the chromatic aberration, the problem that the blue light tends to focus in front of the green and the red light, is what happens in your retina, as I showed you before, is you actually have blue detecting cones that are not as densely packed. In other words, you only have blue cones that are more scattered, because what that does is it works with another filter in the visual system to block out the blue light that would have information with detailed imagery. Because again, the blue light is a fuzzy image, so the visual processing of blue light is different from the red and green, so it removes the information that would relate to fine details because it would be too fuzzy. So again, here again, what we have is a beautiful adaptation, of beautiful engineering to deal with this problem of chromatic aberration. So all of the detailed imagery that's analyzed is purely from the red and green photoreceptor cells where the details can be analyzed. So this is just sort of an example of the machinery. Also what happens is you have what's called an aqueous humor. It's a liquid that surrounds the lens and cornea with just the right nutrients to nourish these tissues. But what happens is you have what's called a canal of sclem, and what this canal does is it removes the fluid. So you have another feedback loop so that it produces just the right amount of fluid to nourish the cells, but if the pressure gets too high, the canal will then remove the fluid to always maintain the right pressure. So you have multiple feedback systems in place to keep this thing functioning properly. Now, if we go to the photoreceptors, what is interesting about the photoreceptors is you have several highly specialized cells. You've got bipolar cells, you've got amacrine cells, you've got horizontal cells, and they all eventually lead into these ganglion cells, which then directly go to the optic nerve into the brain. Well, what happens is these different, this neural network, what happens is the different types of cells interrelate to other cells with different mathematical relationships. So you're having very complex computation in this neural network based on the different relationships of these different cells. And I'll go into what that looks like in a bit. Also, what's fascinating is the photoreceptors face backwards. And people have said, well, that shows that the eye is badly designed because the photoreceptors don't face forward, which you'd kind of expect naively, but they face backwards. So the light actually has to go through all this tissue to get to the photoreceptors. And people say, well, no engineer would do that. But again, that's because they're not engineers. As it turns out, the photoreceptors have to face backwards. And the reason for that is these photoreceptors require more nutrients, more energy. They produce more waste than virtually any other cell in the body. It's very energetic to have to do photodetection. So what happens is this tissue provides the nutrients and the metabolites that are essential for these photoreceptors to operate. So they have to be embedded in this tissue to regenerate a lot of the molecules that they need. In addition, what happens is the photoreceptors have these specialized discs. And what happens in these specialized discs is where you have your photodetecting molecules, your rhodopsin with your retinol. And what happens is they burn out because of all the energy use. So what takes place is because these photoreceptors are embedded in your epithelium tissue, is there's a very complex metabolic process that takes place where the outer discs are actually cleaved off. They're actually broken off, and that allows room for the photoreceptors to regenerate new discs. So if the photoreceptors face forward, they would burn out and you would not be able to see very quickly. They have to face backwards. And also what happens is pretty amazing, is when the epithelium tissue recycles these discs, it actually will take the molecules that detect the photons, re-energize them, reset them, and send them back to the photoreceptor. So that gives you a quicker response time to the light. Also, if we go back to the previous slide, what happens is you have what are called Mueller cells. These cells act like fiber optic cables. They actually deliver light directly to the photoreceptors. In fact, what happens is they have specialized intermediate filaments. You're going to love this. They have intermediate filaments they're actually smaller than the wavelength of light, but they employ a principle called quantum confinement to efficiently transmit the light right to the photoreceptors. So you're seeing advanced nanotechnology. So the photoreceptors are as efficient, and this is something that scientists have realized, is they are as efficient as physically possible. In fact, they can detect a single photon. Now, also, as I mentioned, you have to have the ability to adapt to different intensities of light. They actually have 9 mechanisms of adaptation. So if you're in bright light, what happens is the photoreceptors are adjusted to be less sensitive to the light, and they're less likely to give off signals so that you can see light and tell the difference in like 12 orders of magnitude of brightness because of these 9 separate adaptive mechanisms. So it's just genius design. Here's just a picture of what happens. Is what you have is a very specialized molecule, the rhodopsin. And what happens is when a photon hits the rhodopsin, it hits what's called a retinol molecule inside of this protein. That causes it to go through a change of shape, which then causes a cascade of reactions to take place, which eventually causes a signal to go to your brain that a photon or multiple photons were detected. Now, what's amazing is this only works because of having multiple enzymes, multiple metabolites, a complex cascade to turn a photon into a signal to the brain. But what happens is once this photon changes the shape of this molecule, you need a completely separate system with 3 more enzymes to reset it so it can receive the next photon. Everything is essential at once or it's completely useless. This is how this thing works. Now, that deals with a lot of the structural elements, but what's really amazing is the image processing. Because what you have, as I mentioned, is the retina does what's called pre-processing. It eliminates noise, it gathers basic information. That's then sent to the brain, and the brain has multiple layers of tissue in the cerebral cortex and then an intermediate structure. And what happens is if you study the logic of what's taking place, it works a lot like what we describe as human vision. Because what takes place is if you look at human vision, you have an input like a dog, an image of a dog. Then what happens is it goes through a series of neural layers, which then will extract very simple information like curves, edges, Then what happens, the next layer will create simple shapes like circles. The next layer then starts to look at basic features. The next layer will do things like objects. And then finally, it can identify exactly what you see, like your dog Fido. And what you find is in the brain is you see the same basic logic, that you have the images from the retina that go through layers upon layers of your brain tissue, which then will do things like identify orientation, identify basic shapes. They'll then combine the shapes into more complex features, and eventually they'll be able to identify the object. So when a computer scientist looks at human vision, they can see very clearly what these various stages are doing and why it's doing it. And then what's really extraordinary is what people don't realize is that the retina is considered part of the brain. So even before the images get to the brain, there's incredibly advanced pre-processing taking place. Like in the side of the retina, you have what's called a receptive field center, which could be one photon, it could be more. And then you've got what are called surround fields in these photoreceptors. And then you've got specialized cells, like horizontal cells, that will look at the difference in the signal. So it doesn't just add signals, but it takes the difference. And what this does is it helps to conserve on the information you need. So if you have like an image, which is all the same color, what this does is it shuts off the signals from the photons to send a simpler signal to the brain in a way of compressing the information. Also, this what's called lateral inhibition will determine edges. And then you've got inside the retina other circuitry for determining motion, circuitry to determine if an object's getting closer, getting further away, separate circuitry for determining foreground from background, et cetera, et cetera. But here's my favorite. Here's my absolute favorite. In your retina, you have a bit of a problem because the time it takes for the signal to go from your photoreceptor to your brain could be on the order of, let's say, a tenth of a second, a little bit less than a tenth of a second. Well, if you're playing tennis and you're playing, let's say, against a professional tennis player, a ball could go a whole meter in that amount of time. So what happens is you'll get hit in the head by the ball because what your brain perceives is not going to be where the ball is. But the retina anticipates where objects will be in the future, then sends that image to your brain. So the image that your brain perceives is where the ball will actually be when your brain perceives it. So it's incredibly advanced neural processing, which is pretty amazing. Now, what's fascinating too is one thing we're trying to show with our engineering group is that engineering insights and principles are very powerful in predicting what you'll expect in biology. They're very powerful in helping you understand what you see in biology. And I did a little experiment. I said, well, I studied neural networks, and I know for a neural network to work, you've got to have a training pattern. Any of you that are programmers know this. You've got to send like training data to teach a neural network how to set the right connection strengths between the different neurons. So I said, I bet if I look in the literature, I'll find training data. Guess what I found? Training data. What happens is when you're in development, like your retina will produce spontaneous signals, and those spontaneous signals act as training data to help establish the neural networks in your visual system. And the same is true with your auditory system and your tactile system. So again, you're seeing the same principle in neural networks of training that we use, that the body is using in its system. Now, in addition to that, I talked a bit about the structure of the retina and the photoreceptors, but what's also amazing is that you've got a special optic cup for your eye. So you have 5 bones that produce this beautiful ball and socket type arrangement where your eye perfectly fits in the socket. And what happens is it's well protected, and you have 6 specialized muscles that will allow your eye to go left, to go right, to go up diagonally and down diagonally. So you have these 6 specialized muscles. But what's really kind of amazing is that there's one particular muscle that for it to work properly, it has to come at a very peculiar angle. And there's really nowhere in the skull to mount that particular muscle. So what takes place is in the bone, you've got this little loop made of cartilage, and you have like a tendon that's strung right through this loop as a pulley, so the muscle can pull the eye in the right direction. So just wonderful design of engineering. But here's you're gonna love this. You're gonna love this. Okay, now big problem. because your photoreceptors, if you move your head, what happens is your image on your head will be a blur, right? But your eyes are designed so if I look at an object and I move my head, I stay fixated on that object. So what takes place is you've got special circuitry in your brain that can detect motion, but also what you have in your inner ear is you have these special circular canals or circular ducts, and they're in 3 separate dimensions. And there's a fluid with hair cells, so whenever you move and accelerate, the hair cells detect your motion in 3 dimensions, and that's why you have balance. Some of you more than others. But what happens is the signal from the motion of acceleration is combined with the information from your vision, which allows your head to fixate on a specific object even when you move your head. And that's why you can see a clear image, because of your eyes always being fixated on it, because if you didn't have that, the world would be a blur and high-resolution vision would be completely useless. Now the question then becomes, how do people explain this? For those of you that are engineers, you think, well, that looks like engineering. It looks like design. That seems very clear. But that's not exactly the way everyone sees it. In fact, here's what Charles Darwin said about the eye. He said, "Yet reason tells me that if numerous gradations from a perfect and complex eye to one very imperfect and simple, each grade being useful to its possessor, can be shown to exist, if further the eye does vary ever so slightly, and the variations be inherited, which is certainly the case, and if the variation or modification in the organ be ever useful to an animal under changing conditions of life, then the difficulty of believing that a perfect and complex eye could be formed by natural selection through inseparable by our imagination can hardly be considered real. How a nerve comes to be sensitive to light hardly concerns us more than how life itself first originated. But I may remark that several facts make me suspect that any sensitive nerve may be rendered sensitive to light. In looking for the gradations by which an organ in any species has been perfected, we ought to look exclusively to its lineal ancestors, but this is scarcely ever possible, and we are forced in such case to look to species of the same group, that is, to the collateral descendants from the same original parent form, in order to see what gradations are possible, and for the change of some gradations having been transmitted from the earlier stages of descent in an unaltered or little altered condition. In other words, amongst existing vertebrata, we find but a small amount of gradation in the structure of the eye, and from fossil species we can learn nothing on its head. In this great class, we should probably have to descend far beneath the lowest known fossoriferous stratum to discover the early stages by which our eye has been perfected. In other words, what he imagines is if you look at the different eyes of different animals, you see this gradual development from simple to complex. That's the logic he's using. But the question is, does that actually make sense? Let me use an analogy of why it doesn't make sense. Imagine that you meet a swimmer and they're convinced that they can swim from Tokyo to the United States, California. They're absolutely convinced because they're convinced that there is an island every quarter mile from each other in a continuous set that goes right to the United States. And you ask, well, why do you believe that? Well, because I found the Hawaiian Islands. And because the Hawaiian Islands exist, there must be a continuous series of islands very close to each other going directly to the mainland. That's kind of logic. Now, of course, if that swimmer decided to go on their trek, it would not turn out well for their swimmer. After a mile or so, they would drown. But what happens is this is kind of how the logic goes. That we have, a simple eye spot. We've got the modern eye. And in between, you have an eye with a lens, but maybe it doesn't focus all that well. So in the evolutionist mind, this proves that there's this gradual transition from an eye spot to a modern eye. But the problem is that story only works if you ignore the biology. Because as soon as you look at the biology, the story completely collapses. Because what does it take to have a lens? To have a lens, you've got to have a very complex development process where you have different tissues sending signals to each other, and it has to be controlled by a regulatory gene network. In other words, you've got to have genes that control other genes in this crescendo of activity to direct the origin of that eye. And what's amazing is when a lens develops, what happens is you've got these lens fibers that will elongate, But then what happens is the tissues, the cells actually eject their nucleus and their organelles. They eject what's inside of them, and they fill up with a special crystalline material. They also have very specialized molecules that allow them to link together, to bind together. And they've got specialized channels that allow nutrients to go in and nourish the cells. So if all that doesn't happen at once, your lens is useless. So let's imagine you had 90% of this needed information to go from a cell to develop into an eye. What happens? You end up with undifferentiated tissue. There's a term for that. It's called cancer. It's called glaucoma. So what happens is if you have 90% of what you need to get a lens, but you don't have the last 10%, you have undifferentiated tissue that blocks your vision that's detrimental to the organism. So in an instant, you've got to have all this in place for a lens to form with the right properties to focus the light. So people, I think in the previous talk, asked about theistic evolution. Could God have worked through evolution? In principle, yes. In practice, no. Because even if you were to choose every single mutation, you still couldn't produce this because either it's all there at once and it has a functional lens, or it's undifferentiated tissue which harms the organism. This is sort of the challenge that you have. Now, that's getting a lens. Now let's go from a lens that has low resolution to a lens with high resolution. What's really interesting is you have what's called a box jellyfish. And the box jellyfish has an eye with a lens, but it doesn't focus the light to a sharp point. It simply sort of channels the light into a smaller area. And the question is, could this not evolve into a high-resolution vision? And the answer is it can't, because the neural network of this jellyfish is designed to process fuzzy images. Because all the jellyfish cares about is, let's say, a shark that will swim over it. So all it cares about is shadows, bulk features. So if you were to give that jellyfish a high-resolution lens, so you had a high-resolution image on the retina, it would harm the organism because its neural network is not designed for high-resolution images. So to go from a lens that isn't well-focused by design to a high-resolution image in one instant, you've got to have the neural network for your retina, and you've got to have you have to have the entire network in your brain to process high-resolution images, or it's completely useless. That means you're going to need probably millions of genetic alterations in an instant to go from an eye like this to a high-resolution eye. So the whole evolutionary narrative makes no sense.